Many bacteria have genes that make them resistant to our antibiotics. They can insert these genes into movable genetic elements, which are then transferred to other bacteria, eventually making more and more bacteria antibiotic resistant. But how does the bacterium cut and paste these genes so effortlessly? The secret is the ingenious enzyme transposase. The transposase protein is a homodimer and so has two identical monomers. The N-terminal domain of each monomer contains lysine, arginine, and glutamate residues that allow it to bind to DNA. The transposon is also called a jumping gene and is the gene that needs to be cut and pasted. First, each transposase monomer binds to one end of this transposon, where the yellow residues make contact with its outside ends. Now, the monomers, still attached to the DNA, dimerize, forming a synaptic complex. This loops the transposon, readying it for excision from the donor DNA. This dimerization is facilitated by an alpha helix containing a glycine and an alanine residue. Being non-polar hydrophobic amino acids, these contact their counterparts on the other monomer, causing the dimerization. The formation of the dimer causes the DNA held by each monomer to intrude into the active site of the opposite monomer, forming several contacts between the protein and DNA. Multiple positively charged arginines and lysines contact the negatively charged phosphate groups of the DNA, holding it in place. A small distance away, a thymine base of the DNA is rotated out into a pocket of the protein. Here it interacts with the aromatic residues tyrosine and tryptophan, where the tryptophan ring stacks with the rotated thymine and the tyrosine hydrogen bonds with it. Now that all these contacts have held the DNA in place, the active site can get to work. The active site of each monomer contains 3 acidic residues making up the DDE catalytic triad. These residues coordinate with the key players of the active site, the 2 manganese ions, to hold them in the active pocket. After the DNA is properly fixed within the active site, a water molecule is activated by the first manganese ion. It acts as a nucleophile and attacks the phosphodiester bond between the transposon and donor DNA, leading to cleavage via an SN2 reaction. The free 3' hydroxyl thus formed is stabilized by coordination with the second manganese ion while the donor DNA leaves the pocket. But this cleavage happens on only one of the DNA strands, so how does the entire DNA get cleaved? The free 3' hydroxyl of the cleaved strand now attacks the opposite 5' phosphate of the complementary strand. The strands twist, forming a hairpin structure. This hairpin intermediate is stabilized by the coordination of the oxygen of the newly formed phosphodiester bond and the first manganese ion. This hairpin structure is then resolved by another concerted SN2 reaction, where an activated water molecule attacks the hairpin phosphodiester bond, causing the separation of the 2 strands. The products are stabilized by coordinating the 2 manganese ions. And so these critical cations not only position the reacting groups, but also activate the nucleophile and stabilize the transition states in the reaction. The synaptic complex has now excised the transposon from the donor DNA and has started its journey to identify and capture the target DNA. First, the target DNA is properly positioned within the active site. Here, the free 3' hydroxyl end of the transposon attacks a phosphodiester bond on the target DNA, causing a transesterification reaction again via an SN2 mechanism. The Nic-10s are now sealed by DNA ligases, completing the integration of one bacterium's genes into a completely different organism. Today, transposase is being explored as a promising vector to advance gene therapy, hopefully enabling us to cure a range of genetic diseases. But on the other hand, it is becoming more and more difficult to cure previously straightforward infections, all due to one beautiful but deadly enzyme.